Good afternoon, everyone. Welcome. I'm Jack Van Horn from the University of Virginia, and it's my distinct pleasure to welcome you to this edition of our 2024-2025 Foundations of Biomedical Data Science Seminar Series. Our unique lecture series is made possible through the generous support of the Integrated Translational Health Institute of Virginia, the UVA College of Arts and Sciences, the UVA School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science Seminar Series has been at the forefront of exploring the transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across diverse themes in the biomedical and health sciences. Our selected theme for this year is focused on quantitative approaches in spatial multi-omics for guiding personalized medicine, where each Friday we enjoy presentations from a wide range of leading thinkers about the issues, the promise, opportunities, and the hurdles associated with advanced genomic, transcriptomic, metabolomic, and other omic-related methods as they are related to data science, deep learning, AI, and spatial modeling. Participants for our selected Biomedical Data Science Innovation Lab program will utilize the content from these seminar presentations as essential material in our culminating in-person manuscript and grant project development workshop to be held at the Hutton Hotel in downtown Nashville, Tennessee in June of 2025. We are super excited about this year's program where junior faculty, biomedical, and computational experts will come together to develop novel joint projects on topics related to the quantitative approaches and applications for spatial multi-omics data types and their use in guiding personalized medicine. For today's seminar, I am absolutely delighted to welcome our speaker, Dr. Sean Hanlon from the National Cancer Institute. Dr. Hanlon is the acting Deputy Director of the NCI Center for Strategic Scientific Initiatives, where he provides leadership in the planning, the developing, and implementing of initiatives with a focus on emerging areas of science with potential impact across the cancer research continuum. He is committed to advancing the field of cancer research through open science, technology development, and transdisciplinary collaboration and theory on high-dimensional statistics. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series theme, Dr. Hanlon's lecture today is entitled The NCI's Human Tumor Atlas Network, or HTAN, Generating our Biomedical Data Science Innovation Lab, and as well to the broader scientific community. As always, we are streaming this lecture live and for recording via YouTube. If you're watching as well as alumni are strongly encouraged to submit any questions they have via the chat feature in their YouTube sessions. I'll synthesize these questions and I'll ask them on your behalf during the last 10 minutes or so of the lecture. And with that, Sean, thank you so very much. We are really grateful that you've been able to take the time to speak with us, and we look forward to your presentation.
Thanks so much, Jack, for the wonderful introduction and for the opportunity to share. I'm really excited to share about the NCI's Human Tumor Atlas Network. What I'm going to cover today, I'll spend a bit of time sharing some background and overview of HTAN, and then what we accomplished in the first phase of the program, and what we're moving towards in the second phase of the program, which just launched this past November. And then I'll talk, probably about half the time I'll spend on some of the data and resources that are being made available from the HTAN program to the broader scientific community. So the overarching goal of the HTAN program is to construct dynamic three-dimensional atlases of human cancers. And these atlases integrate molecular, cellular, and tumor tissue composition analyses, including the microenvironment, both the stromal cells and the immune components of the microenvironment. The atlases describe important transitions in cancer, so that can be from precancerous to malignant lesions, from local tumors to invasive and metastatic, and from responsive to therapy to resistance. And in the longer term, we hope that these atlases will enable predictive modeling that can help improve detection, interception, or refine therapeutic choices. So I just want to make some comparisons between the Human Tumor Atlas Network and another large atlasing program that was supported by the NCI for many years, the Cancer Genome Atlas, which people are very familiar with in general. And so TCGA in general had a large number of samples per cancer type, something around 300 to 500 samples per cancer that they were looking at. Where in HTAN, the majority of the studies are smaller. There's usually in the tens to 100 or so for prospective collections. But the HTAN samples are generally analyzed at a much deeper level. TCGA focused on primary untreated tumors, and there was generally limited follow-up clinical data. Where in HTAN, as I mentioned earlier, we're looking at these transitions in cancer, so it's more dynamics, and they include longitudinal sampling. In TCGA, the main molecular characterizations were whole genome, whole exome, and whole transcriptome analysis of bulk analysis of samples. Whereas in HTAN, there's an emphasis on single cell assays and, as relevant to the Bitzel Innovations Lab this year, on multi-omic spatial technologies. Um, and TCGA did have some imaging, mostly pathology imaging data, where in HTAN there's pathology imaging data, but there's also highly multiplex spatial proteomics and spatial transcriptomics data. I'm not going to go through all of this. I think that the audience will be familiar here that there's a variety of spatial omics technologies, but just one thing I want to point out is that HTAN launched in 2017, and the field has really evolved over the last almost decade now in which tools were available. At the beginning of HTAN, many of the both single cell sequencing and spatial technologies were lab generated tests, and there were very few commercial options available. But as people know, there's many more and more commercial technologies available and a wider set. And so many of these different technologies, both in the spatial area and the single cell sequencing area, are being employed within HTAN. And those two buckets of technologies are the real workhorses of the HTAN centers. So the phase two, which we are just starting now, is really building upon the goals and successes of the phase one of the HTAN program. And recently, just in October, there was a large collection of HTAN atlases that were published in Nature. So this is a publicly available set of spatially resolved tumor maps that describe these transitions in cancer for use by the broader cancer research community. There's a Nature cover on the right here with a QR code, which will take you to the collection. I think there's 10 primary atlas papers along with a number of technology-focused papers. And I'll just cover a few highlights of what was included in this Nature collection. So there's one study in this collection that used spatial transcriptomics and spatial proteomics approaches to look at 6 different cancer types and look specifically at the interactions between cancer and non-cancerous cells. And they identified distinct substructures and subclonal regions, which they call microregions, within many of the tumors. And these are smaller spatial regions within different tumors that have different activities and different interactions. For example, there is differences in metabolic state, differences in oncogenic pathway activities, different levels of T cell invasion, and other immune components. there's several studies in the Nature collection that focused on colorectal cancer. And these included two that were focused primarily on the early stages and looking at the early molecular events that drive the transformation from precancerous lesions to frank colon cancer. And there were also two studies in breast cancer, one that used single cell RNA sequencing to profile metastatic cancer cells from 60 uh, patients. And then another which looked at the underlying genetic networks that can define breast cancer subtypes. And this was based on profiles from thousands of cells from each of the different breast cancer subtypes. In addition to these more specific examples from the collection, I just wanted to highlight some common themes that have come across through all of the HTAN publications that were part of phase one. So far, this has included about more than 20 primary atlas publications, more than 50 new analytical and experimental tools, and a total of about 200 HTAN publications and preprints. So some things that we have seen across many of these studies is a coevolution of the tumor and stromal environment as cancers go through these progressions, and we can see that some of these tumor stroma interactions have actually been drivers of progression or resistance. Using a lot of these deep analyses and the single cell sequencing and spatial technologies, we've also been able to discover a number of rare or transitional or plastic tumor cell types. And these cell types are often both conserved across different tumors and they've been correlated with poor progression. And then I think one of the most common aspects that we've seen explored in the HTAN papers is looking at the immune components of tumors. And this is likely helped by the fact that there's a number of good markers for the different tumor types that can be used to identify a range of different immune cells, and they can be explored in various different tumor types. And we've seen things such as immune suppressive signaling circuits being really important for driving tumor growth, and also a role for cancer-associated fibroblasts to act as tumor microenvironment remodelers that impact immune responses to those tumors. And so those are some highlights from the first phase of HTAN. And as I said, these what we've learned through the first phase of HTAN really helped drive the implementation of the second phase of the program. And what we aim for in the second phase of the program is to develop atlases that can help enable the development of new classifiers and risk prediction tools to help identify potential targets for preventative interception, and to improve diagnostic and treatment strategies for advanced cancers. And also importantly, the basic science, basic cancer biology is also important, and these atlases are really key for generating compelling hypotheses to facilitate future research on the underlying biological mechanisms that drive cancer. So this slide is showing the tumor types that were part of phase one of HTAN. And they have the tumor types, and then those with red text, those are focused on and pre-cancer areas, and in dark text where advanced cancers. And you can see that the main tumor types for the first phase were lung, pancreas, colon, breast, skin, and pediatric. And I talked about a few of the findings from breast and colon, as well as a pan-cancer study. And now in phase two, we've really complemented the tumor types that were part of phase one and added a number of new types. And there's some continuing. So both skin and pancreas are there's new centers looking at skin and pancreas in precancer states, but we have new tumor types, including brain tumor, gastric tumor, multiple myeloma, prostate, ovarian cancer, lymphoma, and a number of new pediatric solid tumors. And even in those cases like colon cancer, which we had in phase one, but we now have again in phase two, this colon cancer in phase two is focused on the emerging problem of the rising rates of early onset colorectal cancer. So even where we have tumor types that continue on into phase two, they are bringing some new biological or clinical question to really complement what has already been done. And this slide just shows the structure of phase two of the HTAN program. And so we have 5 centers that are focused on pre-cancer atlases. So focused on this first part here of going from normal to pre-malignant to malignant transitions. We have 5 tumor atlas centers that span the whole range from normal to metastatic to resistance. Then we have a data coordinating center that helps manage the data sharing aspects as well as organizing a lot of the consortium activities and outreach activities. We have a steering committee that helps provide guidance to the overall network. And then we've just launched some new working groups. So we have a pre-cancer atlas subcommittee that's focused on those on issues specific to pre-cancer atlases. They have some different both technical challenges, like that sample sizes are often much smaller than for advanced cancers, and so prioritizing the different types of analyses that need to be done can be different, and also different clinical questions. We have an advisory committee to the Data Coordinating Center that helps guide the priorities of the DCC and what needs to be done to support the network. And then we have working groups focused on analytical methods, visualization tools, translating findings to more clinical space, and then an outreach-focused working group. And again, people should check out the HTAN data portal at this link here, and they find a lot of information about the program and resources. I'll have a link to the DCC on many of my slides. So I just want to talk a little bit about some of the principles around HTAN that we try and keep in mind when we're doing any of the activities that are part of the program. And while HTAN, there's 10 centers that are doing their own research projects, we really want HTAN to support the broader research community beyond those that are directly funded by the program. And so we really want to create a community resource that can catalyze cancer research across disciplines. We also want to use complementary approaches. As I said at the beginning, right, there's been a lot of continuing development of these spatial profiling technologies, and we really want the HTAN community, along with our collaborators and a number of different Atlas focused programs, to be able to work openly and discuss the strengths and weaknesses of the different technologies and where they work well and don't work well, so that we can really make the most use and best use these very powerful tools, but also there's trying to understand which ones work best in which environment is really important. We also prioritize open communication, both within HTAN and with the broader research community. We really want to accelerate science by breaking down walls, striving towards interoperability, Just a few other notes on this. In HTAN, our publication policy strongly encourages members to post their work as a preprints before publication, because we really, again, this is a very fast-moving field. There's lots of excitement both within HTAN and outside of it, and so we want to make sure these findings get out to the research community as rapidly as possible. We also work very closely with a number of other Atlas-focused groups, including the Chan Zuckerberg Initiative-funded Human Cell Atlas. There's a number of other atlases in kidney space and brain that are supported by NIH groups, and so. And then there's the HubMap program at NIH as well that's looking at atlases in normal tissues. And so we try and collaborate both of the investigators that are part of those networks, but then also the program staff that runs these programs also work together so that we can communicate what's working in one program or another, identify opportunities for collaborations. And so, this is and also to support junior investigators. And we have a junior investigator focused Atlas building meeting each year that we try and build a community of people that are working in this space to be the next generation of leaders in the Atlas building environment. And then finally, we have data and resource sharing as another major principle within HTAN. It's a requirement for our success. And we try to encourage aggressive public data and resource release timelines where we're making the data available to the public through our HTAN data portal and as well as our partner groups. And like I said, making publications available through preprints, and also we make our protocols available through protocols.io. So in every aspect, try and make the data and resources developed through HTAN be accessible to the full research community. So I'm going to spend most of the rest of the talk talking more about these data and resources. And so this is a screenshot of the homepage of the HTAN data portal. And either the QR code or the link here will take you to this. And you can see there's a bunch of different options, which I'll talk through a bit. But first, I need to acknowledge the work of our amazing HTAN Data Coordinating Center team. It is led by Ethan Sarami at Dana-Farber, Adam Taylor at Sage Bionetworks, Nikki Schultz at Memorial Sloan Kettering, and Vesta Thorson at the Institute for Systems Biology, along with this much bigger team. And they are they're part of phase 1, and they're again part of phase 2, and they are the drivers of all of these data resources that I'm about to show you. So HTAN generates data from a large number of tumor types and a large number of assays. So on the left here, you can see a bar chart of the data that's available from, I think it's 21 primary tumor sites. You can see that the largest here is in breast cancer, where there's about 1,000 different samples that I have data for. And going down to stomach with the smallest number, that I think if you go live to the website, the number of stomach cancer samples is actually a bit higher. Luckily, the DCC is regularly ingesting new data, but I did not update this to the latest bar charts. And then we also have data available from 30 different assays, and I'll get into this a little bit later. And I think this is one of the challenges, is this creates a lot of work for both our data generators and our DCC team to make sure that we're developing robust data standards for all of these different data types. So the key goal of the HTAN data portal is to make the HTAN data available, accessible, as easily and broadly as possible. And so on the left side of this figure, we have the research centers. So these would represent the different HTAN centers. Um, and they generate their data, they've completed their studies, and they then submit that data to the HTAN data portal at Synapse and ISB, and those will go into some cloud storage buckets and get processed and checked for quality control, etc. But then we need to make it available to the community. And I think as people are probably aware, there are different types of users of data, right? So your cancer biologists, your clinical focused researchers might want a different type of data compared to your bioinformatics or computational or AI researchers. So we try and make these data available through different approaches to be valuable to different communities. And so your cancer biologist that might be interested in using the data to, identify where there's immune cell invasion in a tumor, or what types of cells are interacting when we have resistance to a particular therapy. They might be most interested in the process data and so those process data can be accessed at our HTAN data portal, and they can be visualized and accessed through collaborations with different groups, including we have the Cell by Gene tool that is developed by the Chan Zuckerberg Initiative for visualizing visualizing single cell RNA sequencing data. And so a lot of our single cell RNA sequencing data is available in Cell by Gene. We have a couple of tools called Minerva and Auto Minerva that allow visualization of the spatial transcriptomics and spatial proteomics data. And then there's also links to Google's BigQuery for building cohorts or exploring some of the single cell sequencing data. And then your bioinformatics, computational biologists communities are often going to be interested in raw data and processing data through their own pipelines, or using the imaging data in machine learning algorithms. And so the raw data from HTAN is available through the NCI's Cancer Research Data Commons, including the Cancer Data Service, and also the Seven Bridges Cancer Genomics Cloud, and people can access the data in these environments, and there's be able to do analyses in the cloud where they can bring their own analysis tools to the data. And in some circumstances as well, we can allow for downloading of the raw data for people that want to access the data and process the data locally. So in the next few slides, I'm going to just show you some ways that people can explore and access data on the HTAN data portal. So if you go to the HTAN data portal, there's an explore button on the landing page. If you click on that explore button, it's going to take you to this page here, the portal. And from here, I'll show you a few ways to explore the data. To the top, you'll see that there's a bunch of filters. So you can filter on things like the primary organ or the molecular characterization that you want to look at. And so on this slide, I've filtered for cyclic immunofluorescence. So this is one of the spatial proteomics technologies, and malignant melanoma. And so if I make those filtering selections, I can see here that there's a total of 13 cases and 22 biospecimens and 44 files. And you can see here, these are the SciSif imaging data. You can see the thumbnails of it. And if you were to click on that thumbnail, it would take you to the Minerva visualization tool, so you could explore these files further. Another feature of the portal are these icons at the top here, and these are different visualization tools. And so these first couple of images are Minerva images, are for Minerva visualizations. This is the cell by gene visualization. This is for Google BigQuery. This is our newest tool. It's a link to the UCSC Xena visualization tool. And this is to link out to CBio portal. And I'll just show you a couple examples of how that works. So for the cell by gene, if you click on that, it will take you to the cell by gene tool. And so this will show you a UMAP or t-SNE plots of different cell types. So there's different ways of you're in cell by gene. You can use all of the tools within cell by gene to visualize by different features of the data. And then here is I'll show you a little bit more about the Minerva spatial proteomics and spatial transcriptomics visualization tools. So here's, if you take a link, you'd click on this link that I showed before, and then on the right, you can see a video where you can see that you could be able to zoom in, you could be able to select different markers, and you can be able to explore these really highly multiplexed imaging files in a number of different ways. Okay, so, and I'm going to show you one more feature. It's a newer feature or newly enhanced feature of the portal. That's another way of exploring the data. And so you can see on this explore page, there's a column for publications, and then they have different publication icons next to the different atlases. And so here is the Washington University of St. Louis atlas, and I'll click on this second publication link here, and it will take you to a publication page. And this is the one of the publications from the Nature collection that I talked about at the beginning, that was looking at across looking at the tumor stromal interactions across 6 different cancer types. And if you click on one of these publications like this, you will take you to the publication page, in which you can then see the different data that is part of that paper. So if you had an HTAN publication you were really interested in, and you wanted to dig into the data that were part of that publication, you could find that publication on the publications links, go to the publication page, and then you could see the different data sets that were part of that publication. You could try and explore them on the portal through visualizations, or you could link out to the raw data at the Cancer Research Data Commons and be able to do your own analyses or integrate those data with your data to explore more. And so those are the features I'm going to share about the portal. It's constantly, it's in continued development through the second phase of the program. We adding new features, new visualizations, improving things. So for people that do use it, they do have a ticket system. If you run into any issues, you can submit a ticket for some help, or if you have an idea for feedback, we'd also like to hear that too. Because again, I think making these resources as valuable to the broader scientific community as possible is one of the key objectives of the program. So I just want to talk a little bit more about these data sharing, and then get into we'll talk a little bit about sharing successes and challenges, and then get into some of the ways we're trying to think about how to improve. And so what has been successful about data sharing in HTAN? And so I think one of the, I mentioned we have 30 plus assays. One of the real big successes is that we've been able to engage the HTAN and external communities in developing robust data formats and models and standards for each data type. Um, the way that we do this is our DCC team, often with input from an HTAN working group, will develop the first draft of a data model. And then we'll go out to the community and request comments from all of the different HTAN centers, and revise those. And maybe that has to be a little bit of an iterative process. And then we'll develop a data standard. And what we try as a starting point to leverage existing data standards as much as possible. So for a single cell sequencing data standards, we really leaned on the Human Cell Atlas data standards. For our clinical data standards, we started with the NCI's Genomic Data Commons clinical model as a starting point. Again, one of the other successes has been developing policies that promote rapid data protocol and tool sharing, along with posting of manuscripts on bioRxiv. So we've really instituted this open science ethos within HTAN, and it's taken hold, I think, across the network. But these can be challenging topics, and it is not all, and not everything's been easy. And so, what are the challenges with sharing? So I think, again, having many different assays across many different types of data, single cell, spatial, and bulk sequencing, has been a challenge to, get all of these data models completed and aligned. Developing the best approaches for sharing the spatial omics data has also been a challenge. In particular, because these are newer technologies, really understanding what the community wants, what level of data do they want? Do they want the raw images from these spatial transcriptomics and spatial proteomics data for reuse? Do they want, sort of cell count, cell location, gene count? Is that the data? So we don't know what the community is most interested in. Another challenge is that our cancer research data commons is very cloud focused, and there are not as many spatial imaging analysis tools in the cloud. So we need to make those spatial omics technologies available in the cloud. It's still challenging to link multimodal data, especially I think linking between the single cell and spatial data. And then, sort of meeting the needs. I said we really want to support the computational experts, and we want to support the cancer biologists, meeting the needs of all those users and directing them to the right resources, right? So I showed you, like, there's a lot of different ways to explore the HTAN data. There's different access points to get the HTAN data. Those are all done so that we can try and serve these different communities, but it also can create questions for people about where do they go. And so trying to make sure we document and provide as many sort of tools to help people find the data is important. So one way we've been trying to improve this is getting people to hands-on work with the data and give us feedback on what can make it better. And what we've done is we've hosted a couple of HTAN data jamborees that bring together small teams of folks to work collaboratively using the HTAN data to explore new scientific questions or develop new tools. And the goals of these are to promote the access and reuse of HTAN data, to help drive some new collaborations, develop new methods, and identify gaps and limitations with the existing HTAN data and resources. This slide is just a summary of the two jamborees we've run so far. We ran one virtual one in December of 2023 and an in-person one in November of 2024. We've had almost 200 applications to the two different events, and about 30 different project proposals for projects that could be worked on at the events. We've ended up across the two meetings selected 90 participants to work on 12 different projects. And on the bottom right here, you can see our group from the in-person meeting in November, and you can see up here a breakdown of our participants, and you can see that a large majority have been either grad students, postdocs, or staff scientists, and we've had a smaller number of faculty participants. Um, and the way that these jamborees work, the first day you'll come and we'll give you an orientation of what's going to be expected, trying to set the stage for people to plan appropriately for a 3 or 4 day event. What can you reasonably expect to accomplish? Then we ask the teams to go off and propose, come up with an initial project plan, come back and share that plan with the group so they can get feedback on, are their goals set at the right level? Have you thought about this data issue? You might want to, include this. And then in the middle days, teams go off mainly on their own, working on things, trying to get their projects completed. Then we do come back each day to share updates and where maybe they're stuck on anything and get any feedback. And then the final day, we have them present their final projects. And I have to say that we've been really supported by Alyssa Dillman from Biodata Sage in helping to organize and run these events. And they've been a real success so far. And it's really been impressive to see what people are able to do in just 3 or 4 days with these projects. And so on this slide, I'm just showing some of the project teams we've had across the two events. I'm not going to get into the projects in the next couple of slides. I'll talk about the projects. And then on the bottom, we see some pictures of people working and presenting. One thing I'll just note, in this most recent one in November, we had a fire drill at our event one day, but the teams were so diligent that they didn't give up and they just took their laptops outside and kept on working right through our fire drill. And luckily, we were able to get in on time to complete our final presentations. So in the next couple of slides, I'm just going to show some buckets of types of projects that we've seen as part of these jamborees. And so I'll call these first couple the rigor and reproducibility projects. And so in our first jamboree, we had a publication reproducibility project where the team said, let's look at an HTAN publication, the data that's available in the portal. Can we recreate the results of that paper just from the data and information on the HTAN data portal? And you can see on the left, on this figure, shows that this was a UMAP from the original paper, and here's what they were able to reproduce using the data on the portal. And then a second study I'll talk about in this category was sort of using HTAN data to validate findings from a paper that was outside of HTAN. And they were trying, in this figure on the right here shows some of the work from that team. It was trying to use HTAN data from a pancreatic study to validate some cancer-associated fibroblast subtypes that were identified in a previous publication. We've also had a number of projects that focus on technical challenges. And so I think probably anybody that works in spatial proteomics or spatial transcriptomics knows cell segmentation can be a really challenging step in the analysis. And so we've had 2 projects that have tried different approaches to overcoming this challenge or addressing this challenge. So in our first jamboree, we had a team that tried to implement the segment anything foundation model to improve cell segmentation. And in our most recent jamboree, we had a team that said, well, let's skip this the segmentation step. Can we just use a deep learning approach to do a cell type assignment based on based on the marker expression levels? And then our final bucket of challenges is what I call cancer biology projects. And these are challenges trying to use HTAN data to understand or make new understandings of cancer biology. So we had a project in our first jamboree focused on immune cell infiltration. So they use some single cell sequencing data from one of the projects to look at immune cell information in pediatric, pancreatic cancer. And then in our November Jamboree, we had a team that was using a publicly available HTAN single cell sequencing data to identify regulatory T cells and regulatory T cell subtypes across a number of different cancer types. So just wrapping up on the HTAN data jamborees, some of their outputs. So one of the things, all of the projects are made openly available in GitHub. They're available at this GitHub link here. They've helped us improve the understanding of how people are interested in accessing and using HTAN data. We're also working on a manuscript to highlight both the Jamboree structure as well as some of the project-specific work that was accomplished at the Jamboree that's specifically focused on the in-person one that was from this past November. We're also developing some public projects that were built from the Jamboree team projects that can be used as training tools. And we're working on developing an NCI seminar series focused on HTAN and HTAN resources. We've also gotten really important HTAN data and resource feedback. So we've gotten feedback from participants identifying some specific opportunities for improvement in the HTAN portal and the Cancer Research Data Commons. So, for example, and after the first Jamboree, we got some feedback that it was difficult to go from data in the portal to getting access to that data in the cancer data surface. And so we developed new approaches in order to make that easier to go from the portal to these cloud resources where you can analyze the data. And also the HTAN DCC and CRDC teams, working through these jamborees on their own have developed some potential ways to collaborate, some potential synergistic opportunities. So the last slide I want to just talk about is, again, we try and make these data resources tools from HTAN available to the research community. But I also want to say that the HTAN is not a closed network, and we have ways for people that are interested in joining to become part of the program through the associate membership policy. And so people can apply to become an associate member. Associate members are expected to contribute to HTAN either by generating, sharing, or analyzing data, by jointly developing software or algorithms, developing mutually beneficial resources, protocols, or reagents, and/or coordinating development of standards, formats, or metadata. Associate members can engage in HTAN activities, like our working groups, our annual meetings. They do have to agree to abide by all of our policies. And if people are interested in applying, one, you can click this URL, or you can scan the QR code. But what we need is a letter of support from a current HTAN member. And if that's hard, the NCI team can help facilitate that. We need a biosketch and then a letter of intent from the applicant describing their planned contributions to HTAN. And so these applications in general come from a PI level person, but can include people from their lab as being part of HTAN as part of their associate membership. And with that, I am just going to say that, I talked a lot about all of the work that's going on in HTAN. All of the research is done by the extramural investigators who, have been really engaged with this program. But then all of the coordination activities that I talked about are not just done by me. We have a big team at the NCI. We have the DCC folks that I showed on a previous slide. But these are the NCI folks that have been engaged with in particular engaged with HTAN Phase 2, who are my wonderful colleagues who I have to acknowledge for all of their hard work. And so with that, I am happy to take any questions. And
Sean,
yeah.
oh, I was going to say thank you so much, and thank you for providing your contact information there in case anyone has any questions, and checking you out on LinkedIn as well. Thanks for that. I I have a couple of questions, and my first one is entirely selfish. Is that this is a fantastic resource, and it just looks like so much work has gone into this, and it is exactly on point with some of the data types that we hope to have people utilizing. I'm wondering if there's any way and anything we can do to help showcase these data and this resource at our in-person BDSIL event in June, and maybe get you a bunch of new young investigator user people. How could we do that?
Yeah, I mean, certainly the data resources are generally open. We could find somebody from the DCC team to present. I potentially could present. So happy to find a way to do that. And I agree with any ways to. We're really excited to find any ways to get people access to these tools. And so yeah, I'm happy to be engaged with that or find people to connect you with to that would be good contributors.
That'd be super, and I'll reach out to you under a later time to talk about the technical details of that. But for some of the richness of the data that you've shown across all the different kind of organ systems where you have cancer data, what do you feel are some of the kind of the data science needs? I know you have a great team that's helping to develop these tools, but do you think that there's some areas that some of our junior faculty, bioinformaticians and cancer biologists and other micro or omic type folks, along with their data science partners, might attempt to take on as projects?
Yeah, I think that's a great question. I mean, some thoughts that came to mind are integrating across. Is it possible to integrate across data sets generated by different groups, or different technologies? I think that's one question. I can, in the spatial proteomics, let's say, maybe there's only a handful of markers that are the same between different, um, you know, a study in breast cancer and a study in colon cancer. Is that enough? Or what's the number of markers we'd really want to have in common to be able to do some meaningful integration across those? That's an area that just frankly, there hasn't been that much bandwidth. And there are a lot of technical questions within HTAN community about how can we or can we integrate across compare across different studies, either in the spatial or even in the single cell sequencing. A lot of the single cell sequencing data, each group process that data through their own pipelines. Would we need to reprocess it through a common pipeline in order to compare it? Or are there some comparisons we can do as is? How do we even check that? Those are some things that come to mind that I think we could really use some thoughtful bioinformatics, computational insights into.
And is data visualization something you are concerned about?
Yeah, I think data visualization also remains a challenge. Again, sort of are there ways we can link the single cell and spatial data if I have it from the same patient in a visualization tool? Um, I definitely think visualization, and maybe that also gets to visualizations that will support the sort of the the more cancer biology or more biological focused person being able to gain access. I mentioned TCGA in one of my early slides, and that's been a great tool for the people focused on the more fundamental cancer biology questions, because I can go in and say, what I'm interested in this gene, what cancer types is it, mutated or upregulated or downregulated in. The HTAN data is more complex, and so sort of ways to visualize the complex data that makes it accessible to less computationally, focused communities, I think would be a great asset.
Yeah, I like the notion that this data is really complicated and that just saying that, something was, the signal went up or the signal went down, it's just more nuanced than that. And being able to describe that visually may be more helpful for people who are non-specialists or people from adjacent fields who are trying to get their heads around it. And that probably includes people who are data scientists. So it's sort of
Yeah,
like,
yeah,
it's a chicken and egg question in terms of
yeah,
visualization and then a further analysis, but you can see how they might go hand in hand.
absolutely, absolutely.
Well, I know that you have got an open access policy for these data, and you're encouraging people to come to get that data. It sounds like you have to get HTAN sponsor to kind of give you, vouch for you. Well, once you've got that,
Well, so wait,
okay, correct, correct
yeah,
me if I'm
yeah,
wrong.
yeah, yeah. So the published, most of the data is openly. You don't have to be any part of HTAN. You just the data is made available. There is some the raw imaging data is controlled access, so people would have to make a request through dbGaP to access the controlled access data.
Okay.
Um, the becoming an associate member that really just allows you to come to our face-to-face meetings, join our working groups, be a little bit more sort of in depth, maybe hear some of the stuff more
Okay, I see.
pre-publication. That's not a you don't have to do that to access the data. That's just a way if you wanted to contribute more directly to the program.
Oh, great. Thanks for clarifying that. I mean, I'm involved with several NIH related databases, and you have to like get some, a university signing official to sign some sort of thing, and it goes through your grants and contracts, and it ends up being kind of a whole thing
Yeah,
in order to get access to that data. But it sounds like, at least for versions of the
yeah,
HTAN data, you don't have to do that, which is wonderful because it's really kind of a barrier to have to go through a bunch of documentation, get it
yeah.
signed by people and stuff. But that's fantastic.
Yeah, and so that's why, like for our HTAN data jamborees, we focused on those open access data sets so that we didn't have to, deal with any signing officials or whatever for any of our participants in that, because they were mostly junior investigators. That it's even harder for those folks to get that type of agreement in place.
Well, we would be super excited about, featuring this, promoting it, encouraging people to draw from it. I mean, this is exactly the type of data that we would hope that people would want to look at. And while we would not necessarily be like one of your jamborees, we have elements of the jamboree slash hackathon slash project development kind of thing involved in our BDSIL activities. And so it'd be fantastic to one, showcase some of that data, have some of our data science attendees as well as our kind of biomedical attendees taking a look at that data, asking questions about what can be done. And then my final question is, ordinarily, in ordinary times, we encourage these teams to look for various funding sources to be able to support projects they have. And I realize that things are a little different now, but are there any HTAN related, or do you anticipate any? Well, maybe you can't even say that, but would there be, say, R21 type grants that people might tend to target?
So what I can say is that NCI has an ongoing program called the Informatics Technology for Cancer Research, or ITCR program, that
Okay.
supports development of cancer-related bioinformatics tools uh, across the life cycle. From It has an R21 funding opportunity, a U01 funding opportunity, and a U24 opportunity. So, the ITCR program supports really widely known tools like CBioPortal, but it also supports new algorithm development through its R21, new tool development through the R21.
Those would be fantastic to point our folks to and encourage them to take a look at that. Again, we realize that things are a little uncertain right now, but we want to give people at least a target to aim for should it be available. So, Sean, we're at the end of our hour, and gosh, it was fantastic to learn more about HTAN and all the different things and how it might be directly in line with our activities here coming up this summer. And so, thank you so much for sharing this. I'm going to reach out to you under separate cover to talk a little bit more about how we might feature this resource and engage with some other of the HTAN investigators to help provide some more insights into how to utilize these data for people's projects at our event this summer. So thank you so much.
Thank you. Really exciting for the opportunity and appreciate it.
Thank you so much. Everyone have a wonderful weekend. We'll see you next time.